Hi, welcome everyone. Thank you for joining this webinar. We're so excited to have you here. My name is Kate Crochelle. I am one of the marketing managers here at iMotions, and we have a really exciting Path to Publication webinar for you today, featuring our awesome longtime client, Anne Bjertup. I'm going to introduce her in a second, and we're going to talk about her awesome research with iMotions that has spanned many years into researching mothers with affective disorders, which she will tell you a ton more about, so I don't want to get into it too much, but just a quick housekeeping note before we get started that I will give a little intro to iMotions during this webinar, then Anna will present her PhD research and the publications attached to that. We will also have an interactive Q&A during this webinar, so if you have a question at any time, please feel free to put it into the chat that you can see on your screen into the little question box. We will be queuing those up and answering them along the way in written format, but then we're also going to have an interactive Q&A at the end. So any questions for Anna that you have about her research, please put it into the question box at any time, and we will make sure to ask it. And also, if you're wondering, this webinar will be recorded, and all of the participants who have registered, whether they're joined now and you guys are seeing it live, or if they've just registered but they can't attend live, then you'll receive the recording tomorrow. So in 24 hours, it'll be in your inbox. So no worries if you have to jump off at all. You'll get it tomorrow. So yeah, we have Anne here. Welcome so much. I'm so happy to have you here to present your really interesting research. So Anne Juul Bjertup is a PhD and postdoc researcher. She has been affiliated with iMotions, or she's been an iMotions client since 2017. So it's been a long time, and she actually started doing her PhD research together with iMotions. She has a master's in psychology, and then she is now affiliated with the NEED group, which is in the mental health services department of the University of Copenhagen's capital region. It's a little bit confusing, but if you live in Copenhagen, where she and I do, it's in the capital region, which is our like big healthcare network. So she's in the NEED group there, and she can tell you a little bit more about what that means during the webinar. But we're so excited to have you. So thanks for joining, Anne.
Thank you. Thank you for the kind introduction. Thanks for the invitation.
Great. So I'm just quickly going to give 2 minutes of an overview of what iMotions does, just so you get a background of what we're all about, and then you'll kind of have that as context if you aren't familiar with us while Anna describes her research. So we have worked as a software company for over a decade. We're actually coming up on 16 years. We were founded in 2015, and we have offices all around the world. You can see here. Our clients execute advanced human behavior research. What does that mean? It means that we are working with biosensors, the hardware that we are resellers for, and we also integrate natively with our software, all of these different biosensors. Basically, biosensor research is a way of sort of tracking and analyzing the physiological signals that our bodies give to reveal the sort of underlying emotional content, and also the attentional and cognition content kind of below the surface. So anything from physiological arousal to visual attention, from eye tracking to galvanic skin response, which is a measure of sweat activity from the skin, and a bunch of other biosensors that we all integrate with, and we timestamp them, and we synchronize all of the data from a variety of these native partners that we integrate with. And then we are able to basically streamline that biosensor research that you might want to do into cognition, attention, emotion with your participants. So, like I said, we synchronize and integrate all of those signals into one platform, and then we also give you the opportunity to analyze that data and export it. So whether you want to work directly in iMotions to do these nice visualizations, for example, if you're working with eye tracking, you might want to do some areas of interest or gaze mapping, but you can also export that data into other systems so that you can work in, for example, MATLAB or other data analysis systems that you're used to. So yeah, researchers across the academic and commercial space, you can see some of them there that we work with, but I don't want to dwell too much on that because that's not the point of this conversation. This is about learning about Anna's research. and all the really innovative stuff she's been working with in the field of psychology. So let's take it away. We're just going to have kind of an informal conversation, kind of back and forth, but I would love to start by asking you, Anna, how did you first get interested in this type of research, and also how did you hear about iMotions?
Yeah, so I've been interested in this human behavior research since before I started studying psychology. And then for my master's thesis, I did my first small empirical study on emotional cognition in pregnant women with and without affective disorders. I did that in collaboration with Professor Camilla Moscoviak, who I've worked with ever since. So here my interest in this group, and especially the emotional neurocognition, really began. And in the meantime, during my master's thesis, Camilla got funding for the PhD project on mothers with affective disorders that I was then very qualified for. And at that time, she had been in contact with iMotions, and some of my colleagues did a pilot study with on eye gaze and facial displays of emotions in patients with bipolar disorder. And this was right at the time when I was starting up my PhD. And then Camilla and I thought it would be super interesting and very useful to include these biosensors in the PhD study on these mothers' emotional cognitive processing of infant stimuli, to sort of get this more comprehensive understanding of how mothers respond and process to infant signals. And this was also very relevant, since you could say that real life mother interactions are multimodal, and both mothers and infants, they communicate with, for example, their facial expression and the gazes. So by including these measures and different biosensors with the computerized paradigms, we could sort of get closer to measuring the processes that actually occur in these real life interactions.
Yeah, super interesting, because it seems like this is sort of still kind of an innovative look at measuring these kind of in the moment processes that I would say that a lot of us kind of quote unquote take for granted, but actually using biosensor research to analyze those in the moment interactions. I just think it's really interesting, and it must have also been for you, as it sort of like triggered this kind of research question for you, that you wanted to end up doing a whole PhD on it. So can you tell me a little bit more about, specifically on your PhD study, what was the initial goal with the research, and just kind of talk about the aim and how it kind of took shape?
Yes, sure. So let me just start by with a little definition and a little bit of background for those who do not work with affective disorders in psychiatry. So affective disorders include both bipolar disorder, where patients experience both manias and depression. And then we have unipolar disorder, where patients only experience depressions. And unipolar disorder is often just called depression. It's the same thing. So the overall reason that we're interested in investigating these processes in mothers with and without these affective disorders is that the disorders are associated with some neurocognitive biases in how emotional information is perceived, recognized, and responded to. So, for example, the most consistently reported is that depression is associated with negative cognitive biases, for example, in interpretation of other people's emotional expressions as facial expressions. And also mothers with postpartum depression, we know that they are more withdrawn in interactions with their own infants, and they sort of show a more flat and negative facial expression towards their own infants. And another thing is that these disorders are very heritable. So children of parents with these disorders are at an increased risk of developing the disorders themselves, but also more generally at risk of adverse development. And from attachment theory and developmental studies, we know that the early emotional environment is and the parental responses are vital to the child's psychological development. So Therefore, the overall question is how emotional neurocognitive difficulties associated with these disorders may be related to how mothers respond to their infants, and if that sort of can explain some of this increased risk in the children. Yes, and you can just change slides, please. Thanks. So the specific aim of the study was to provide an empirical investigation of the neurocognitive and psychophysiological responses to infant stimuli in these mothers with affective disorders compared to a control group, healthy control group, and how this was associated with mother-infant interactions, what we can actually observe in real life, and with infant development. So the PhD includes these different levels of scientific analysis, including both neuronal processes, the physiological responses, the cognitive, and the relational in terms of what is going on between mother and her infant. And under the cognitive level of analysis, we use these different biosensors to measure, for example, attention and more pre-conscious or automatic processes, yes, of visual attention and spontaneous facial expressions in combination with more conscious ratings and interpretations of these kinds of stimuli.
Right, and I want to just add quickly here, I think it's really important the level that you have on the top here of the rating and the interpretation of this infant emotion, because what we like to advocate at iEmotions too is that, yeah, biosensors can tell you a lot of the stuff that might not be as apparent in a, for example, a self-report. If someone is responding with their words of what happened in a moment, or sort of attaching a behavior to those words, you can capture so much, but you also need to reinforce that with that actual interpretation and rating, because those two data points kind of work simultaneously and can complement each other. Can you just explain a little bit more about how you actually did that rating and interpretation on top of the biosignals?
Yeah, so for example, the actual task of some of the paradigms was to rate how negative or positive they thought an emotion was, or how negatively or positively they were themselves affected by watching this. So there was this interpretational level of trying to interpret what the infant is signaling, for example, where they had to think about and rate it on a rating bar.
Right, exactly, and that is also a capability of iEmotions, just putting that in there, that you can do rating questions during a study. But yes, let's maybe talk a little bit more about the methodology around the data collection, because I kind of hinted, and you did as well, about how you're actually layering these different signals into the aim of the study. But can you just tell me a little bit about your methodology behind this PhD research?
Yes, so the study builds on existing research that use these computerized paradigms where, as we just talked about, participants, for example, watch some infant face images or videos, and then are asked to interpret them or respond to them somehow. So we did this, but then also added the biosensors, for example, to reach an insight into the more automatic and pre-conscious processes. We for example included the eye tracking, as you can see in the figure here in the middle. Sort of shows how this is an infant video, and this sort of displays how the eye tracking keeps track on where they gaze and fixate at the infant. Then they also their facial expressions were also recorded as this measure of their how positively or negatively they were affected by watching this infant stimuli. And we also measured the galvanic skin responses, which is this physiological arousal in response to watching some emotional stimuli. And so I recruited these mothers with and without affective disorders, and they're around 4 months old infants. And then I visited them in their homes on the first day, and then they went through all these affective cognitive tasks, and also filled out a bunch of questionnaires, and I assessed infant development, and I did this video recording of mother-infant interaction that were later on coded by someone blind to the group status. Yes, and then at another day they went through an MR scan where they watched images of their own and an unknown infant face in the scanner. Yeah.
So why was it important to layer in all the different criteria? Like you said, the live coding of the sort of the coders were blind to the study, and then also sending them through an MR scanner. What were those like methodological tactics? How did that add to, or sort of like complement the biosensor data in this design? What was your goal of having these, like, you know, parallel data points to look at? Was it so that you could really isolate the control group from the groups that you wanted to study in terms of the affective disorders?
Yes, so the overall aim is to see if there's group differences, which we hypothesize given the affective neurocognitive difficulties that are associated with these difficulties, but including all these different levels of measures sort of brings more pieces to the puzzle, which I'll talk a little bit more about when discussing the results. And the mother-infant interaction, for example, I think it's really important to, in terms of validating the neurocognitive and biosensor results, to associate it with what's going on in real life. Does it even associate, or how can we understand these measures? Because it's very complex, and it's difficult to interpret just with one measure, I think.
Yeah, exactly. And on that note, like, if you are considering using biosensors for your research, no matter what your use case is, especially if you're sort of like newer to this technology and these methodologies, you might be tempted to just gather a ton of data points, right? Just like put all the sensors on the moms and see what happens, but you really want to be careful and critical about what actual data points you want to bring in and how you can analyze those, and like Anna said, you know, really isolate what you're trying to accomplish based on your hypothesis. So that's just a little like best practice signal that just remember, more is not necessarily better. It's really just about crafting it towards how it fits your research. Yes. Okay, so let's move on a little bit. Yeah, so you mentioned we can talk about the data conclusions and the results, but I kind of touched on this a little bit, but I'm really curious about how you did actually draw the conclusions using these biosensors, and you had mentioned the control group and then the group with the affective disorders. I would love to hear just how what results you got, basically.
Yeah, so in one of the articles, if you can just change slides again. Yeah, in one of the articles, we looked at these computer tasks and associate them with the mother-infant interaction and infant development. And so if you change the slides, we found that when looking at mothers with affective disorders in just one group of affective mothers overall, they show fewer galvanic skin response peaks when watching infant emotional videos, and they also gazed less at this infant stimuli. And we interpreted these findings as indicating that these mothers were somehow less vigilant and attentive towards the infants. And the figure to the right here shows that the less mothers gaze toward the infants in the video, the lower their own infant scored on the cognitive scale of the development assessment. Exactly. So this, of course, does not say anything about the causal mechanism, but I think it's still interesting that the tendency to maybe look away or avoid infant stimuli somehow is related to your own child scoring lower on a cognitive scale. And then if you change the slides again, we also investigated these mothers with bipolar and unipolar disorder separately, and found that mothers with bipolar disorder, they had a slightly positive bias in their responses to infant cries, that they rated a little less negatively. And interestingly, they also displayed more positive facial expressions when watching a video of an infant crying heavily. So this is sort of a very misattuned and not congruent response to the infant emotion, which is also in line with other literature. And if you change the slide again, I have some figures of the mothers with unipolar disorder. In contrast, they displayed this negative cognitive bias with more negative facial expressions when listening to infant cries, and they gazed less at intense infant happiness, but slightly more at sadness, which is again in line with previous findings that show that mothers with depression have a tendency to avoid intense infant emotions. Yes, and if you change the slide one more time, we correlated all these measures across each other, and we found that so overall the less time spent gazing at infant videos and faces, it correlated with both lower infant cognition, but also with lower scores on a communication scale, and with less dyadic reciprocity. So this dyadic reciprocity is a measure of mother-infant interaction and how much both mother and infant are involved in sort of a give and take interactions, how much they participate in joint activity and respond appropriately to each other's signals. So this was lower for the mother-infant diets, where mothers looked or gazed less at infant stimuli on a computer. And another interesting finding was that the more of these sort of misattuned positive facial expressions toward an infant in distress correlated with less maternal sensitivity in interactions with own infants. Yes, so as I just said before, I think the results from these different levels of analyses, including all these biosensors, can be interpreted in light of each other, and add more pieces to the larger puzzle, because this is a very complex field, and it's difficult to interpret when some behavior is beneficial and appropriate and when it's more adverse, and we need also a theoretical background to interpret what is appropriate and what is adverse. You have to look at it in a context.
Yeah, definitely. It's super interesting, your findings here. And when you say previous literature, have there been other studies that have been performed using biosensors that have found similar things that you've come across? Or were you really like trying to underscore research using other methods that kind of underpins this finding with biosensors?
Yeah, so there's really not been very much research done in this area, and most of it includes only one or two measures, and it's typically only ratings. But there is a few studies using, for example, eye tracking that show some of the same things with the avoidance of intense emotions, and and also actually the studies that used galvanic skin responses or some measure of skin conductance, they find very heterogeneous results. But I think that also very much depend on the context. And what was different in our study is that we investigated mothers who were in remission from the symptoms. So, and this is the first study ever doing that. And the reason that we did that is that these disorders are episodic. So you have more depressions or manias, but in between these, there are large periods where you don't experience any symptoms. But you're still considered having this disorder because you have the vulnerability even though you can actually live for years and years without experiencing a new depression if you're very well treated. And so I think previous studies have typically been done on mothers with acute depression, where these depressive symptoms are much more, they affect the responses much more than these sort of subtle the subtle responses experienced in remission. But the reason that we were interested in investigating it in remission is that if we can show that there are these subtle neurocognitive differences even in remission, we show that the child is going to expose to this all the time, in daily interactions, and not only in acute depressive episodes. And in terms of the clinical implications, you really have to work with the relation and with the interpretation and responses to the child's emotions, and not only focusing on treating a depression, because the risk that the children are exposed to does not necessarily go away with the depressive symptoms.
Right. That's super interesting to think about and remember that, yeah, this has really broad potential implications. And yeah, doing those interventions can actually be beneficial not only for the mother but the child in their long-term development. I think that's really interesting to note, and that actually brings us to our next point, I believe, which is what you're going to talk about when you talk about what you're working on now. But was there anything you wanted to add about this PhD research specifically, like in the publication that you had, published as a result of this PhD work?
So I just added 2 more slides regarding the results. So the other article we published was looking at the fMRI data, and here we found that the mothers with bipolar disorder, they had reduced response in the left dorsolateral prefrontal cortex when they watched their own compared to an unknown infant. And they also had this positive bias when they rated images of their own infant's distress. So they interpreted this as less negatively than healthy control mothers did. And they also rated their own emotional responses to watching their own infant's distress as less negatively. So they were less negative affected by it. And that correlated with this reduced dorsolateral prefrontal cortex response when they watched own infants. So this is just, again, another result that sort of point in the same direction, and it's in line with the sort of less appropriate or less regulated response. Yes, so.
Right. Yeah, and super interesting too about the sort of positive bias with your own child, that the kind of pictures, like you give some examples here, they kind of go on a scale from distressed to happy, right? So if that is, you can say that the one on the very left that's kind of covered a bit would be, I'm guessing, the most distressed, right? But it's kind of a subjective measure as well. But it's super interesting to see that the mothers who have these affective disorders actually rate it less negatively than the control group. Yeah.
And we actually had a group of people not affiliated with these babies at all rate all of these images just to see if they rated them differently. So if there was sort of objective differences in the intensity of their emotions, and there were no differences in that. So the results can't be explained by infants of bipolar mothers just showing more positive emotions. Yeah.
Right. Yeah. And just to me also, that's kind of a surprising finding, the sort of like a positive reaction that the mothers with bipolar had, because that's not necessarily, like, if you're thinking about like the perception of what bipolar, how it can manifest, you wouldn't necessarily conclude that. So it's a super interesting finding that you've found. Yeah, just based on sort of the general population, what you would associate with having a bipolar disorder.
Yeah.
Yes.
And it is super difficult to interpret, and it's very preliminary results, but there's some other research on some different stimuli also indicating this positive bias, and maybe it's more related to some more broad emotional cognitive difficulties and difficulties in understanding and recognizing and really understanding expressions in general. Yeah.
Yes, definitely. And again, to bring it a little bit back to what iMotions can do and help you out kind of in when you're assessing these findings is because, Anna, you used facial expression analysis and eye tracking and galvanic skin response too, you had those three sort of data points. I mean, not in the fMRI part of the study, but you have the three data points to sort of say, like, we know exactly when this moment is happening and correlating it to the actual stimuli, looking at these images. And you can kind of say, all right, so what is the facial expression analysis doing right now? And you have that in the moment data. So it kind of gives you like a sort of leg up, or it sort of boosts the robustness of your findings because you have those three data points that you can correlate. Would you agree?
Yeah, exactly. And I think it's actually important to mention that, because we also found the reduced galvanic skin responses, and we thought, oh, maybe that has to do with them since they are gazing less at the actual stimuli. It might be therefore that their galvanic skin responses are also reduced, but those measures actually didn't correlate. So it's and I think that's important, because then it's not explained by them looking away, because it was also the galvanic skin responses were also lower when they were actually looking at the infant stimuli.
Yes, sorry, I was a little trigger happy with the the slides there. Super interesting. So yeah, I'm conscious of time because we want to give time for questions. So I want to ask you what you're working on now.
Yeah. So so the study I did as my master's thesis was actually published, and an interesting finding was that negatively biased rating of infant cries during pregnancy predicted who got postpartum depression after birth. So if you change the slides. So this is just a little bit of explanation of the things that I'm looking at now, because so the figure to the left here shows women who later on got postpartum depression. They are the blue dots to the left. So they had more negative ratings of infant cries during pregnancy. And we have actually found this pattern again very recently. So the figure to the right is preliminary results from an ongoing, almost finished study, where again, pregnant women were asked to rate some infant stimuli, and those who were more negatively affected by it during pregnancy, they were more likely to develop postpartum depression later on. So these figures just look so similar, and I think that's very interesting. So so if you change the slides again, the emotional cognitive biases, like these negative biases in how you respond to infant cries, they may both be a risk factor for the infant during the interaction, but also mark a risk for depression later on. So based on these findings, and based on the findings from the PhD study, we wanted to investigate if we could intervene and modify these emotional cognitive biases, and if it's possible to modify these already in pregnancy. And so we have recently conducted a pilot study where we tested the feasibility and the effect of such a sort of prenatal affective cognitive training, which is what is depicted in the bottom of this figure. So pregnant women at high risk of developing postpartum depression went through this training, which, for example, includes some attentional bias modification training, and here we used specifically an eye tracker that continuously analyzed where these women gazed while they watched some videos of infants, and if they looked away from the infant's face, the screen got more blurry and red, which would remind them to look back at the infants, and then the screen would get clear again. So this was quite a sophisticated paradigm that was programmed in PsychToolbox in MATLAB with much help from iMotions, and I'm extremely thankful for the help that I got with realizing this idea. And so another task we also included was where pregnant women were instructed to use their facial expressions to sensitively mirror and interact with infants in videos by making facial expressions that were sensitive to emotions displayed by the infants. And again, this task, so we used biofeedback again, and the task was run from MATLAB with iMotions open, because we used the affective FDX algorithm that continuously processed these action units of facial expressions, and then evaluated whether the facial expression was sensitive or not, and then the participants got feedback based on that feedback that they continuously had to incorporate in their responses during the task. Yes, and this image here just shows how we determined which action units were involved in facial expressions, how we evaluate them as being correct or incorrect. Yes, I think it's very interesting that so we show different facial expressions towards infants than we do towards adults, and one study actually specified which specific action units are involved in infant-directed facial expression, infant-directed facial expressions. Yeah. So if you just change the slide again, we just submitted the first results from this pilot study on whether there were any changes in affective cognitive responses in these high-risk pregnant women, and we found that after the training, they felt less frustration when watching infant distress, and they also rated their own emotional response to infant cries a little less negative at follow-up than at baseline. And if you change the slide again, the pregnant women also gazed more at infant distressed faces after the training. So this attentional bias modification might have worked. And they displayed more infant-directed facial expressions toward infant stimuli. But although this was only found at the immediate follow-up and went away at the delayed follow-up. And lastly, if you change the slides again, these pregnant women at high risk of depression, they also rated infant happiness, so images of infant happy faces and neutral faces, they rated them more positively at the follow-up visits, so after the training. And for most of these tasks, a control group of women at low risk of postpartum depression, they showed no changes in these affective cognitive processes. So those who didn't go through the training. So based on this pilot study, we are now starting up a larger randomized controlled trial of this intervention, where we'll, of course, have a high-risk control group and many more participants than in the pilot study. But I think it's some promising results that we got from the pilot study.
Yeah, really, really amazing research you're doing. I think it has huge implications potentially. So I'm really glad that you're moving on to the next phase beyond the pilot study. And I wanted to highlight also what you said about using the MATLAB scripts and using feeding it through iMotions, because this is the kind of stuff that we as a company can actually help you with. We have a lot of PhD researchers ourselves on staff who have tons of experience with these tools, and we can help you consult on your study, basically. So this study that Anne had created has, for the pilot study, did have some sort of custom coding in it. So I just want to highlight that. I want to be transparent. This is not something you can kind of get out of the box with iMotions, but the paradigm that you've created is just like, it has such huge implications for actually, like you said, training these mothers who might be high risk and kind of trying to minimize that risk for them and for their infant, that we're so excited to be involved in this research. So we just want to say thank you for all of the work with iMotions you've been doing. But yes, so I think we're rapidly running out of time, but I want to just ask you a last quick tip about actually getting to the publication stage, because this is the title of the webinars we have, is tips about the publication process. And while I'm asking you that, I want to name for the audience that if you do have a question for Anna, please put it into the chat now so we can make sure to get that answered for you. But yeah, what can you tell us about actually getting this published, this research, the several research studies that you mentioned? How would you suggest like going about the publication process when you know, from sort of the sort of end or closer to the end of the process, but also thinking about it maybe as you're designing your research, where you want to publish? Can you give us any tips?
Yeah, so we'll first of all, doing a very sound study with high quality methods and well substantiated hypothesis, and a strong design and sufficient sample size and so on is of course key to getting published. But apart from that, then I think more practical, on a practical level, you can very early on in the process start thinking about which journal you want to submit to, for example. And for this purpose, I think that the journal finder is a really useful tool. And this is something that I think most of the larger publishing companies like Elsevier and Wiley, for example, they have that. So if you just Google journal finder, then these different tools for different journals show up. And in these, you can type in typically the title of your manuscript, maybe an abstract, the scientific area, and some keywords, and then it will match with different journals. And I think that's actually, it actually does some quite good matching, at least in my field of research. And another thing, and the next step is to look into the aims and the scope of the journal to see if your manuscript matches these, and also always be aware if you'll have to pay a fee. It's very annoying to sort of edit your manuscript to fit the scopes of some journal, and then you figure out that you have to pay a large fee, and it won't be able to do it anyway. And yeah, always read the author guidelines very carefully. It will make the review process much more easy.
Yes, great tip, thank you. So journal finder, you can plug in keywords and you can find the journal that might match your studies. So that's great, thank you. So we have about 15 minutes left, and we already have some questions coming in. So please, if you have questions for Anna, please put them in right now, and I'm going to queue them up for this Q&A. So before we do that, I do have a question for you, the audience. If you would like to hear more about iMotions and even maybe connect with Anna about her research yourself, we obviously will share the recording from this webinar tomorrow, but that'll be right in your email inbox. But if you have other ways that we can follow up with you, we would love to know. So I'm just gonna launch a quick poll, and while we do that, you can answer that. It'll just take 2 seconds, and while I do that, we can actually ask some of the questions from the audience. But here I'm going to launch this little poll, and you should see it on your screen now. And if you don't, let me know. But and we have a question about funding. So you have a lot of experience actually in funding for these research initiatives. Can you tell us a little bit about like what you would suggest in terms of like writing grant applications? Do you have any ideas of that you can share with the audience of what has worked for you in the past?
So I think it's important, sort of the same things as when you're writing a very good protocol and your research design. But then for the funding, really focus on why this research is needed, based on existing research. Why is it needed, and how can it make the world a better place? That emphasize really that, but also it's very important to present realistic methods and realistic time frame and a realistic budget, and it's important that it shows from the application that you really thought every little detail through, because I think what they often look at is is this even realistic, might even more than is it needed, the study, but yeah, really, is it realistic? And then also do a thorough research into which foundations fund your type of research. Yeah.
Yes, yeah. So those grant proposals should be as thorough as possible, right? And like you said, using the funding bodies at your disposal to find out who's actually distributing money, and what the time frame of that is. Like, you always want to work backwards, right, from the time frame, from the deadline, all the way back to how long it's going to take you to write the process.
Yeah.
So yeah, thank you for that. That's really helpful. So it looks like the majority of people have answered the poll. So I'm going to close it now. And thank you for answering, everyone who did. And I'm just going to turn off my camera, or sorry, turn off the slides as well, just so we can chat and ask the rest of the questions with the 15 minutes that we have left. So yeah, the next question I have is from Katie, and this is about your PhD research. Would there be any potential differences between looking at and interpreting your own child's emotions in real time versus the static pictures of other people's babies? So there's actually, I think, a few questions in there. It's not only the real-time question versus the sort of static pictures of whether it's your baby or it's someone else's baby, but also the question of real-time versus images generally.
Yes.
Do you have any?
Yeah. So this is a very, very good question, and such an important point, because in terms of validity, this would maybe be the ideal thing to do, but it's more difficult. Then it would be a different measure. So you could, of course, ask mothers while interacting with the infants to rate some different scales. In another study, we actually used a scale on that is called My Emotions When My Infant Cries. That taps into this. So there would be expected to be some differences, although I think they are quite comparable, given research that has combined or associated these measures from computerized tasks with measures from real life interactions. Yes, and actually a very recent study that validated these sort of computerized measures of responses to infant cries, and how mothers responded to their own and interpreted their own infant cries, and they correlated. So those that were more negatively affected by cries on the computerized task and rated them as more negative, the same pattern showed at in interactions with their own infants and in interpreting their own infants' responses, but much more research that sort of combines these two measures are needed.
Yeah, so plenty of research capabilities for you in the future. This material is rich for your studies. Yeah. Okay, great. I have another question from Kirsten, actually 2 questions. Great questions here. So number 1 is, the research discussed was only about mothers. Could it translate to fathers or other caregivers? That's the first question. I guess we can talk about that first. And then there's a question about longitudinal research, but let's take the first one first.
Yeah, yes, I should actually have mentioned that, because I think it's so important to focus on the fathers as well. And the only reason that we only look at mothers is that the attachment theory is basically built on research with mothers, and it's just very recent that more and more research with fathers has come. But that research shows that some of the patterns are the same, but there are also some differences in how mothers and fathers are responding and interacting with infants. And most research actually indicate that the sort of tuning into infants' emotions and all of this starts a little earlier for women. They're also affected by physiological changes and all these kind of things affecting the brains during pregnancy. But then the fathers tune in, and their brains and hormonal system actually also change once the child is born, and the more they interact with them. And of course, it's very important to say that the fathers are as important attachment figures as mothers are as well. So ideally, we would have investigated both mothers and fathers, but for practical reasons, we focused on the mothers.
Right. Yeah. And I love in the question too, Kirsten asks, it's about fathers or other caregivers as well, right? Because sometimes you don't have that sort of like stereotypical nuclear family unit with a heterosexual couple of a mom and dad. Yeah. So again, possibilities for more research.
It's actually the same with the adoptive parents. They also show some neurocognitive changes actually once they are experience-driven changes in brain responses and processing of the infants.
Yeah, exactly. Okay, cool. And then the second part of the question is, can you say more about related research looking at longitudinal effects of reduced reciprocity and sensitivity of caregivers with affective disorders on child development?
Yes, so most of this research, I think, is done by Professor Ruth Feldman, who has done a lot of also synchrony studies, and there are evidence that this reduced mother-infant reciprocity predicts later more adverse child development in terms of both cognition and also in emotional development. Yes, I don't by heart really remember the very specific results, but there are longitudinal studies examining this and overall showing that reduced maternal sensitivity and reduced reciprocity is associated with later adverse child outcomes.
Yeah, really, really interesting as well. Cool. I'm very aware that we are running rapidly out of time. So if you do have a question still that you'd like to ask Anna, now is your chance to put it in. So I have one question from Tien. And you touched on this briefly when you mentioned, that you had specifically recruited mothers who were in remission from their sort of episodic, depressions and bipolar, phases. And Tien also writes that bipolar states shift, right? So did you follow how the change in the mother's level of arousal, valence, and mood state influence how they respond to their babies? So if you could just outline again kind of why you chose the segment of the population that you did and how you could measure the things you did.
So, okay, if I understand the question correctly, actually these mothers, they stayed in remission as we investigated them, and we only did it in 2 time points, so I wasn't really able to investigate sort of the shifts, but we also tried to correlate some of these measures with mood state and also anxiety level and stress level, and some of them did correlate and some of them didn't. I actually don't really remember which did, but overall it made sense, so that more depressive symptoms correlated with more negative bias, for example. Yeah, I don't know if that was really a response to the question.
No, I think it makes sense. So So Tian, if you want to clarify if that doesn't make sense to you, feel free. And then we have a question also here about how did you actually recruit the mothers, like practically? How did you actually find these people to not only the 76 mothers that you mentioned in your PhD study, but also the more recent pilot study?
Yeah, and this is, I think, the most difficult part of doing clinical research. It's the recruiting phase. It's just such a bottleneck. But I have some very valuable collaborations with practitioners and clinical midwives and sort of departments at a hospital. There's one department with special care for vulnerable pregnant women, vulnerable families. I have a good collaboration with those. Then they ask all their patients if they want to volunteer and participate in this study. So yes, good collaborations with clinicians is key. And then I actually recruited the patient mothers from all over Denmark because we had to do something. So I sometimes ventured off to Jutland and made some home visits very, very far away to get enough participants.
Well, great efforts are necessary for this type of research. So great job going to the ends of Denmark for the sake of science.
Yeah.
Yeah, and actually just on the note of collaboration, that made me think, because we have a question asking about like the funding programs and sort of how you can make this research kind of sustainable, because it is expensive, right? So not only if you're actually looking for a grant, but kind of sustaining the research long term. I just wanted to mention in response to this question about funding programs that if you can find collaborations with other groups who are doing similar research, like you can sort of make the case for a shared lab, right? That, you know, in your case, Anna, you're working with the NEED group, but you're also affiliated with the University of Copenhagen and the Capital Region. So like there are possibilities of sort of like creating that network. Can you just talk briefly for 2 minutes about how you could see that facilitating this type of biosensor research due to the expense?
Yeah, and that's also, I think, related to the difficulty with recruiting these patients, these mothers or pregnant women, because once you have them, you just want to get everything out of them. So measure with all possible tools you might think of. And what we actually want to do in this upcoming larger RCT is having a collaboration with a professor at the Department of Psychology who has got some equipment on, has got some FNIRS equipment that's sort of a functional neural measure, because that's super useful to measure infants' neural responses. And you can actually, so what we're going to do is measure both mothers and the infant's neural activity while they are interacting with each other. So this is one of the longitudinal measures that we will include. So because they have that very expensive advanced equipment, we make this collaboration, and the other way around, other labs can cooperate with us with this with our iMotions equipment. Yeah.
Great. On that note, we are out of time. So I just wanted to thank you again, Anna, for taking the time to talk with us about your research and telling us the process from start to finish. And I think I speak for our entire audience that we all really got a lot out of this. So thanks again. And again, if you would like to receive the recording, as long as you're here right now, you're going to get it tomorrow. So it's going to be in your inbox. And if you have outstanding questions that we didn't get to, please email us at marketing@imotions.com, and we will put you in touch with the appropriate person, whether if it's Anna, or if you want to see the publications specifically, I'm happy to send those links over. And if you want to connect with her, she's around. So thank you so much, Anna.
Thank you so much.
Okay. See you.
Bye.